بسم الله الرحمن الرحيم، والصلاة والسلام على أشرف الأنبياء والمرسلين نبينا محمد وعلى آله وصحبه أجمعين. أسعد الله صباحكم جميعا بكل خير. نرحب بكم في المركز الوطني لأبحاث وتطوير الزراعة المستدامة باستدامة في سلسلة اللقاءات والمحاضرات العلمية التي يستهدف فيها المركز نشر الوعي العلمي والمخرجات الأكاديمية والبحثية من الباحثين. سيكون بإذن الله اليوم الحديث عن آفاق علمية للزراعة باستخدام المياه المالحة، ونستهل هذا اللقاء بكلمة من سعادة الدكتور عبد الرحمن بن عبد الله صقير مدير عام مركز استدامة.
بسم الله والصلاة والسلام على رسول الله. الزملاء والزميلات المتحدثون والحضور، السلام عليكم ورحمة الله وبركاته. من المعلوم للجميع أن مصادر المياه الصالحة للاستخدام البشري والزراعة محدودة على مستوى العالم وشحيحة في بعض المناطق، وأن القطاع الزراعي يستهلك ما بين 80 إلى 85% من المياه مقارنة بالقطاعات الأخرى. ومعلوم أيضا أن عدد سكان الكرة الأرضية في تزايد مستمر، ويتوقع أن يتجاوز 9 مليار نسمة بحلول 2050، وهذا يتطلب زيادة إنتاج الزراعة أو المحاصيل الزراعية بنسبة 70% لتلبية والوفاء باحتياجات البشر. هذه الحقائق تبرز ضرورة الاهتمام بمصادر المياه غير التقليدية في الأغراض الزراعية، وهي أي تلك المصادر عديدة ومتنوعة، ومنها المياه المالحة غير الصالحة للأغراض الأخرى ومياه البحر، مع الأخذ في الاعتبار الآثار الجانبية لاستخدام المياه المالحة على التربة ونظامها الحيوي. إن تطويع البحث العلمي والابتكار لتعظيم الاستفادة من المياه المالحة في القطاع الزراعي دون الإضرار بالتربة وخصائصها من المجالات المهمة ذات الأثر الاقتصادي والتنموي الكبير، وهو يسهم في تحقيق التوازن المنشود بين الأمن الغذائي والأمن المائي. وتتنوع الطرق والأساليب لتحقيق ذلك، وهذا ما سيتطرق له الزملاء المتحدثون في ورشة اليوم. ولأهمية هذا الموضوع ودورها الحيوي في تحقيق الزراعة المستدامة، يسعدنا في مركز استدامة أن نستضيف نخبة من الخبراء والباحثين لإلقاء الضوء على ما يمكن أن يقدمه العلم والابتكار في هذا المجال الحيوي البالغ الأهمية. نشكر المتحدثين على مشاركتهم ونرحب بالجميع، ونبدأ على بركة الله.
شكرا دكتور عبد الرحمن. سيكون بإذن الله جميع المحاضرات موجودة على بث مباشر من خلال حساب مركز استدامة عبر تويتر، فيمكن نشر الرابط إذا في أي أحد مهتم. العنوان الأول نبدأ فيه بإذن الله هو التحديات والفرص الحالية لنباتات العشبية الملحية الإيليروباس لترورس لتعزيز تحمل المحاصيل للملوحة نحو الزراعة الملحية المستدامة، مع الدكتور عفيفي من جامعة الملك سعود.
السلام عليكم ورحمة الله. بسم الله الرحمن الرحيم. Good morning every person. I would like to thank Dr. Sugir, Dr. Moin, and all the staff of Istidama for inviting me to assist this important workshop. My speak will be is entitled Current Challenge and Opportunity of Halophyte Grass Aleuropis littoralis to Enhance Salt Plant Tolerance in Crop Toward Sustainable Biocidal Agriculture. What is plant stress? If we can say very simple definition, plant stresses are defined as any change in environment conditions that cause the less production than ideal plant genetic potential. These stresses can effectively, seriously the total production and the yield per hectare. Why is salinity a real challenge for sustainable agriculture? We know that the presence of salt in the soil or irrigation water can inhibit some growth water uptake, inhibit nutrient uptake, and the result is nutrient imbalance and poor root growth. Also, it can affect physiological mechanism of the plant, and finally, it can affect production. How we mitigate to this stress? So there is a first approach is to increase crop production on currently usable arable land by improving plant productivity under stress condition. This implies what? A major breakthrough in crop breeding by identifying stress tolerance gene from extremophile and then introducing them into the traditional crop. But due to the shrinking of arable land by salinization, depletion of freshwater resources, we came up with second strategy. This strategy consists of shifting the focus of agriculture for the production to alternative crops such as domesticated halophyte. This can be achieved in saline area such as marginal land or in semi-arid and arid area where we use saline water for irrigation. This is possible because we know that almost 1% of total flora in the world, either monocot or dicot, are halophyte. So the halophyte are the salt-tolerant plant that thrive and complete their life cycle in the presence of more than 200 millimolar salt in the soil or irrigation during evolution. They have developed different morphological, anatomical, and physiological strategies to proliferate in high salt environment. We have obligate halophyte, facultative halophyte, and habited and different. These halophyte belong to different family of the plant, but mainly concentrated in Chenopodiaceae and Poaceae. So what is the application of halophyte? We can use halophyte as photoremediation tool, phytodesalinization. It can offer forage and bioenergy plant, wood and fiber, balancing carbon cycle and reducing global warming, serve as food source for a human, serve as model plant to test mechanical and resistance to salt stress, coastal protection, serve as a source of medicinal plant, and finally, they can be a potential source of salt resistance gene and promoter that can be introduced in the crop. And finally, they can be a potential source of halotolerant bacteria or fungi that have a biostimulant activity. So this will imply what? It is important for us, especially in our countries, to do a bank of this halophyte. This database of halophyte can offer to us information about plant type and life form, ecotype, maximum salinity tolerance, the presence or absence of salt gland, photosynthetic pathway, antioxidant, secondary metabolite, compatible solid habitat, and economic use. This database can serve in the selection of halophyte for sustainable biosaline agriculture. In the second part, after this introduction, I will present the main research activity that we do in our group. We are working on a plant, what we call Aleropis. Scientific name is Aleropis littoralis. It belongs to grass family, Poaceae. We call in Arabic lacresh. This plant has a very good potential to thrive in harsh conditions like salinity, like Sahara to fix sand, waterlogging area, drought area, and we have elaborated a strategy of two objectives. The first objective is to isolate gene and promoter from this plant and to generate marker-free transgenic cereal elite cultivar tolerant to drought and soil. We are interested in wheat, rice, and also we can be interested in sorghum and maize. The second objective is to harness the rhizosphere of this plant to isolate plant growth microorganisms that can have biostimulant activity. So I will start with the first objective. We have developed a strategy of 5 phases. First of all, we identify gene by SSH Sedena library, RNA-seq, genome sequencing. Then we validate this gene in model plant. And after validation, we can pass to the target plant like wheat and rice. And we do the tolerance assay. And our final goal is to do the pyramiding of the gene in our target plant like cereals. And for the second objective, we have 4 phases. We isolate the microorganism, evaluate the biostimulant activity, identification of this microorganism using molecular biology tools, and finally, we want to formulate biostimulant product that can be used in formal agriculture or hydroponic system. So, Aleropis littoralis is monocotyledon plant, halophyte, adapted to arid region, has a C4 photosynthesis, and unlike other grasses, it has a small genome estimated at 349 megabase. We see here in this photo, they have small chromosome, 10 chromosomes per haploid genome, less in size than rice and wheat. This plant have developed system to thrive in saline condition, either by the presence of salt gland, bicellular salt gland. These salt glands are dispersed in the surface of leaf, and when this collected cell, plenty with salt, it can be exploded and excrete salt, which is toxic to the cell. This crystal of NaCl can play another role. They reflect light and they decrease temperature on the surface of the leaf, and this, by the way, decrease the evapotranspiration. So that's why this plant and have C4 photosynthesis. In addition, this plant have adaptation in the root. We see here very well, when increase the salt, we have longer root, and we reach the longest root at 300 millimolar, which is almost 18 grams per liter salt. But after this point, the plant will behave as glycophyte, as wheat. When we increase salt, we have short root, short leaf, and more we increase salt, more this negative effects is important. So we have started our story by developing hydroponic system for Aleropis to construct Sedena library from root, leaf, under salt stress. Then we are using SSH Sedena technology and macroarray to isolate differentially expressed gene under salt stress. With this technology, we have isolated 492 differentially expressed gene. These 400, after blast with the database, we see that 68% have known function, 12% has undefined homologs sequence, but interestingly, 20% of this sequence are considered as new sequence because they don't have hit with the database. This work was published in Gene. Okay, in a second step, we did a transcriptome sequencing of this plant using 454 technology and Illumina. This is next generation sequencing. And we have developed 4 Sedena library, control leaf, control stressed leaf, root control, root stressed. And after in silico analysis of this transcriptome, we have identified 38,456 differentially protein, and it is almost the entire transcriptome of this plant. So after performing differentially expressed in silico of the gene, we have identified 429 differentially expressed gene. That means upregulated or downregulated gene in this plant. So as a conclusion for this part, Aleuropis is diploid. It has 10 chromosomes, perennial, monocotyledons, halophytes, C4 photosynthesis, salt-secreting, rhizomatous plant, and have a small genome, 349 megabase. So this plant, all these factors make this plant a reservoir of promoter and gene involvement in response and adaptation to environment stress. So we have identified different genes. They have function in protein like LTMP1, LTMP2, protein implicated in regulation of the gene like SCRECO, MIB, transcription factor, LSRG1, and ILSAP gene, ADP ribosylation factor. I will concentrate my speech on this gene, which called the zinc finger protein, because we have reached the end of our strategy. That means we have introduced this to wheat and rice and evaluated the tolerance. So this gene encodes for proteins, 159 amino acids, each one copy in the genome, and it codes for zinc finger protein with a domain AN20 in the N terminus of the protein, and zinc finger AN1 domain in the C terminal. We have performed RT-PCR, and we have demonstrated that this gene is induced by salt, osmotic stress, heat stress, cold stress, and even growth regulator like ABA, abscisic acid, and salicylic acid. So this gene is cloned in binary vector and introduced in agrobacterium. We developed a transgenic line of tobacco, and by southern blot, northern blot, and western blot, we have demonstrated that this plant produced the protein zinc finger. After that, now we should evaluate this plant against salt stress. So at germination stage, we see here that transgenic line overexpressing this gene germinated very well under salt stress, under 200 millimolar salt, that means 10 gram, 11 gram almost of NaCl, and also on osmotic stress and ionic stress when using lithium chloride. So the overexpression of this gene in tobacco plant enhance the germination under ionic stress, NaCl, and osmotic stress. Then at growth stage, we have demonstrated that the transgenic line developed very well either by for root or vegetative under osmotic stress comparing to wild type, and even under salt stress also, and double of these stress. In addition, the transgenic plant recover very well after heat stress for 2 hours at 55 degrees and freezing at -20, and they recover very well. But however, the wild type, they are unable to recover after this stress. And now, final step is the evaluation of stress under greenhouse. And we see here that under control condition, no difference between transgenic 3-line of transgenic line and wild type. However, when we do stress at 350 millimolars irrigation water, we can show here that the plant can continue their life cycle, produce seeds. However, the wild type is unable to produce seeds. The same thing under drought stress when we irrigated by 25% of field capacity. And here we see the growth of the root of transgenic line under drought and salt stress. And here we see that the root of wild type completely necroses under stress. So this is published in Plant Molecular Biology. So after this encouraging result, when we validated in model plant, we have decided to pass this gene to economic plant, which is wheat, durum wheat. For me, I consider that our region especially where there is drought, we should be interested in durum wheat better than bread wheat, because the quality of durum wheat produced in the harsh condition in our region is better and better than those produced in Europe. So we have selected a variety Karim, which is sensitive to drought, sensitive to salt. We know very well that durum wheat is very recalcitrant to regeneration under in vitro condition, not like bread wheat. And the transformation is also very difficult under in vitro. So we have developed a clever idea as an embryo transformation. So what we want to produce transgenic wheat without selection marker, the antibiotic. So what we call marker-free transgenic line. So we have cloned this gene under a promoter from rice. It is actin. And we have injected to here germinated seed in the meristematic zone, because in the meristem there is some cell that can become a spike and produce seeds. So we hope that from this pool of seeds we can find transgenic seeds, and we have selected by PCR for the presence of the seed. Now we should prove that this transformation is stable. So we have used southern blot, and we selected the 3 lines with different number of copy, and northern blot to prove the presence of RNA, and western blot to prove the presence of protein. So our method for agrobacterium transformation of durum wheat produced stable transgenic wheat, as demonstrated by southern blot, northern blot, qPCR, and western blot, and they are marker-free. This is the important point. There is no antibiotic gene here. So we have, after that, we have multiplied this seed until we have T4 homozygote seeds, and we have did the evaluation. We see very well that under control condition there is no difference by phenotype of this transgenic line and wild type. However, under 100 millimolar and 150 millimolar, this is almost 5.8 gram NaCl in water, and this is almost 11 gram. So we see that the germination is clearly different from wild type, and even under 200 millimolar, the wild type are unable to germinate it. However, transgenic durum wheat are able to germinate it on this harsh concentration of salt. In the same way, for vegetation, here we see the growth of the plant under 150 millimolar, and here the wild type, the difference is significantly clear. So after that, we did perform evaluation of this transgenic under greenhouse condition, and we see here that under drought stress or salt stress, under control, we have no difference between the seeds of wild type and transgenic. For this line, the wild type under drought, they produce seed, but they are bad quality, not good fillet. But here, this line produce acceptable quality of seeds. For salt, wild type is unable to produce seeds. For transgenic line, it produces seeds, and they are acceptable quality. So, as a conclusion, we have able to produce transgenic marker-free, that they are salt tolerant and drought tolerant, and they overexpress only one gene from this plant under the control of promoter from rice. This is published in Molecular Breeding. After that, we have did the same work with rice in collaboration with CIRAD Montpellier, and we have obtained almost the same result. And now we think خلاص? So I just pass quickly on this. So this is the promoter of this gene, and the main important thing result is that the promoter of this gene is induced by salt. It is age dependent. It is not expressed in in root. And the application of this promoter is very important, since it is not expressed in the seeds. It can be used for producing pharmaceutical substance in plant for a human or animal in the safe way, because it is not expressed in the seeds. We are now doing, just I pass very quickly, 2 minutes. We are working on the sequencing of the whole genome of this plant. This is very important, and we have succeeded to do this with a project from MPST, funded by MPST. So we used the next generation sequencing, and we did a short read by BGI, SEC, and PacBio. We have now in the lab a server. So we did the assembly of this genome, and almost we have 2,110 contigs, which cover 308 megabase and represent 88% of the genome. And we have identified 40,650 genes. So this is, can be, we have do the chloroplast genome of this plant. And we have identified 62 genes which are implicated in abiotic stress, as described by previous publication. But interestingly, we have now 533 genes which represent by AST and match with unnamed or hypothetical plant. And this is a very good reservoir to work on in the future. So if we want to do the study of this gene, but we wish that we find fun, and our dream is to establish a platform to validate this gene and to finish at the end to do the pyramiding of this gene, either by using conventional breeding methodology or biotechnology and molecular breeding assisted. Just final by this is the biostimulant that we have isolated from the rhizosphere of this plant, and we have isolated 6 strain, and we proved the stimulation of the growth. And when using cell culture feed rate, we can reduce by 50% the chemical fertilizer input in hydroponic, and we can have same growth as full nutritive solution, or even more growth when we do 0.5 nutritive solution, additioned with this biostimulant. Just conclusion end. So as a general conclusion, we can say that the plant has remarkable ability to grow under high salinity and drought condition. It can be used, one, improve and stabilize eroded soil. It can be used as animal feed in form of forage, to be good alternative candidate as bioenergy crop, especially if we develop it in hydroponic, like this photo. The draft genome is very important because it can help us to identify gene and promoter from this plant, and to understand how the mechanism of salt gland will excrete this salt, how the root architecture is adapted to salt. And finally, the harvesting of this plant growth. I would like to thank all the group, Dr. Abdallah Dous, Dr. Emmanuel from Sirat, Donaldo, Thierry Legavre, and all students which participate in this work and give either by PhD or master degree. And finally, this is just for you. We can isolate 2 homolog gene, but one very near to homolog gene, but one give tolerance to salt stress and another give tolerance to drought stress. I thank you very much. I'm sorry.
Thank you, Dr. Afifi. Now شكرا دكتور عفيفي. نظرا لضيق الوقت فنضع الأسئلة إن شاء الله في نهاية المحاضرات بإذن الله. So now we'll go to دكتور مارك. الحديث عن تطوير وتقديم أنظمة زراعية تعتمد على المياه المالحة للمناخات الحارة. Dr. Mark from Center of Desert Agriculture, KAUST. Dr. Mark, do you hear us? Dr. Mark? Dr. Mark? Yeah.
Yes, hello.
Hello. Good morning.
Good morning. What
Okay.
is happening? Is anything happening?
Yeah, please, thank for joining us in this lectures. So please, can you start now for 20 minutes?
Okay.
Yeah, please.
Okay. So, okay, good. With pleasure. Okay.
Thank you.
Share screen. Right. Can you see my screen now? Hello?
Still,
Hello, can you
yeah.
hear me?
yeah, still not see the screen. Yes, now.
You can see my screen now?
Yes.
And a presentation here. Okay, excellent. All right, thank you. I'm not quite sure what sort of audience I'm talking to, but I hope everything is clear, and I'll do my best to give you a little bit of an introduction to some of our work on developing and delivering some saltwater-based agricultural systems. And I've added in the extra bit here 4 hot climates. I think that's an important extra dimension for, not always, but often when it's salty, it's also hot. And we certainly in this region, in the Middle East, need to be coping with both. Some background information very quickly. A lot of our food is produced under irrigation, and a lot of the water that humans use is for food production, but all groundwater resources that are being used for agriculture are being depleted, and clearly this means a large fraction of the world's food is being produced from unsustainable mining of groundwater. And as groundwater is depleted, it also gets more brackish. Nevertheless, there's still huge amounts of water in the world, mostly in the sea, and it would be wonderful if we were able to unlock seawater and other water we currently cannot use to try to contribute to food production. We can do this to increase yields in brackish water systems and also to decrease pressure on freshwater reserves that are used for irrigation, as I was describing in the previous slides. If we're going to unlock something, we need a key. And at least from the perspective of our own research, the key is genomics, which is turbocharging genetics, and hand in glove with phenotyping and new phenotyping technologies using computer vision and machine learning. We have huge opportunities for serious innovation, which includes, very excitingly, very big opportunities to unlock wild germplasm where there is vast genetic resources, including vast amounts of salinity tolerance and heat tolerance for the future. And we can use this type of approach to either make existing crops more salt tolerant and get plants which are very salt tolerant and make them more like crops. One example of the former is work I did actually now quite a few years ago, where we had a durum wheat which had a much higher ability to maintain low shoot sodium. It actually turned out to derive from an old cross with a wild relative from the Middle East, and led to, in saline environments, very big increases in yields, big increases in salinity tolerance. This work was described many years ago, in particular in a paper in Nature Biotechnology, and what is now happening is that work is now being adopted in Bangladesh, where this gene for sodium exclusion, which was there from a natural cross, not from genetic modifications, but from a wild relative, is now being introgressed into Bangladeshi wheat varieties and giving good yield advantages in moderate to high salinity environments. So that's just one example of how there's a real increase in salinity tolerance and is now being delivered in the real world. The other approach is to get salt-tolerant plants and domesticate them. And one example of this is quinoa, which is a crop for marginal lands. It can grow in very saline environments in its original habitats in South America. It also has huge potential as a crop that both can be very high yielding, such as here on the Tibetan Plateau in southwestern China, and it's also highly nutritious. So it provides a seed which can deliver a very well balanced amino acid profile for human health. It's also, I think, a crop that's got a lot of tolerances, most notably missing heat tolerance, I should emphasize, but it's got very strong drought tolerance and salinity tolerance. So it's a crop which I would suggest is suitable for the future world, if only we could increase some of its domestication properties and increase its heat tolerance. So towards this, what we've been doing is collecting germplasm, a wide variety of germplasm for as many sources as is legally, I should emphasize, possible. We've got very good quality genomes now, and we actually resequenced 1,000 different accessions, but there's a lot of duplication in the collections. So we now have 600 original distinct accessions for which we now have very good genomic information, which now enables, if you like, the turbocharging of genetic improvement of the plants. And hand in glove with the genotyping, we've been developing and actually doing phenotyping on quite a large number of traits across quite a large number of sections through the years, different sites, using both traditional, if you like, methods of phenotyping, and I should say there's nothing wrong with that. Things related to yield traits and plant growth morphology, such as height, branchiness, and so on. And we've also been phenotyping using drones and a range of hyperspectral style cameras, trying to develop cheap methods for phenotyping these plants in a wide range of environments, which then allows you to use statistical genetic studies, what are called genome-wide association studies, where you're associating particular phenotypes with a particular location in the genome and plotting a probability of a particular location in the genome with the effect on a particular trait. And from that you can quite quickly move to both breeding programs where, rather than having to phenotype all of the plants resulting from a cross, you can just genotype them. And it also allows for the academic studies to identify the basis for flowering time, day length, sensitivity of flowering, plant height, and so on. All these traits which really are important for fully, if you like, commercially domesticating these plants. And another example I'm going to introduce, by the way, I did say that quinoa does have high salinity tolerance. It can grow very well in brackish water, 1/4, 1/3 seawater. It still has good yields. But there are a relatively small number of plants which are more spectacular in that they're able to grow in full seawater and produce some interesting products that could be of use for plants, for humans, And one of these examples is Salicornia. Salicornia is a plant which grows in full seawater. It thrives particularly in about 1/3, 1/2 seawater, but it can still fully complete its life cycle in full seawater and also quite high temperatures. So we've been looking at actually many species of Salicornia now. Traditionally, people have been growing Salicornia bigelovii, which is a species from southwestern US and northwestern Mexico. But it's an autotetraploid and suffers inbreeding depression, and it's very, very difficult to use as a breeding material. So it's good as a starting material, and it has large seeds that are oil rich, but it doesn't have its prospects for significant genetic improvement are very, very limited. So we're actually taken 3 steps backwards and gone back to looking at plants that are diploid. So Salicornia bigelovii is an autotetraploid. There's no grain crop on the planet that's an autotetraploid. In fact, there's really only 2 crops that have autopolyploidy, and this is very important because they do suffer inbreeding depression and genomic instability. The 2 crops are potatoes and strawberries, neither of which are grown for producing seeds. So for a seed-producing crop, we need to have either allotetraploids or diploids. So we've gone backwards, if you like, and been searching for diploid Salicornias, and there's local species. We've been collecting these and starting to develop inbred pure seed stocks, and these collections, I should say, are ongoing. Some of these plants are coming from really extreme locations, such as this irrigation drainage lake in Qasim in central northern Saudi Arabia. And we've now collected plants from all over the world, annuals and perennials. It doesn't have to be an annual. I know most crops are annuals, faster speeds of breeding, better disease control, better agronomic management. But in growing plants in seawater, I don't think it necessarily needs to be an annual. But we are focusing generally, I have to admit, on annual species from several countries and starting to collect quite a few different species and then starting to characterize them. This is Salicornia bigelovii, in particular found in northwestern Mexico. I should also say that having a local Salicornia is really good for regulatory reasons. We're able to materials, get permits for doing that more easily than getting export permits to, for example, remove Salicornia brachiata legally out of India. That's very difficult. But in the meantime, we're also starting to get information on the ploidy levels of these plants, which is very important and very surprising, sometimes the results. For example, this Salicornia europea, grown from, collected by me near my house here in the south of France, is actually tetraploid, but is claimed to be by the taxonomists to be Salicornia europea. But Salicornia europea is usually diploid, and the diploids and tetraploids, they're isolated reproductively. They have to be different species. So there's a lot of work still to be done on the taxonomic semantics, if you like, of these plants. But in the meantime, we're starting to get information on the ploidy levels and get information on the genome sizes using flow cytometry and then genome sequencing. So at the moment, we've got 2 species with very high quality genomes now developed, and we're very close to a similar level for 3 other species of quinoa. Do so much work with quinoa, with Salicornia. So just to introduce you to the concept here of what we're doing to try to domesticate a wild salt-tolerant plant, and I should say we're doing all of this now with the Salicornia. You get your germplasms, and this is non-trivial because of the legal complications in collecting internationally. So I should say we are focusing on the Middle East, where we are. We then get the genomics, which provides the foundational backbone for enabling a breeding program to move quickly. We are developing inbred lines. We're learning to grow them and phenotype them. We're doing quantitative trials that are properly designed for breeding programs and functionally characterizing the plants. So we're trying to identify the genetic basis for important traits. So important traits are related, of course, to yield and yield-related traits. Also agronomic features such as the way the plants branch, how short they are. And importantly here, I'm not sure I said this early enough, I apologize, but Salicornia produces a seed which is very rich in oil. So those properties of the oil need to be characterized chemically. It turns out that Salicornia has a wonderful quality oil, fantastic for cooking. It's also got lubricant properties that can be used in machines, but we need to characterize those oils and make sure in the process of developing a high-yielding genotype that it's also got good chemical properties for the oil. We need to genotype these plants, of course, so we're learning how they work and their characteristics. We need to genotype them, just as I described for the quinoa. We need to use genomic information with the phenotyping information to enable the development of high-quality germplasm for the delivery of the output, the seeds. Finally, I still have about 5 minutes, I think. I hope, Mr. Chairman. We have here a final introduction to some of our work where we're trying to address high temperatures and really, this is a very, very important topic, not just for our region. Obviously, it's critically important for our region, but globally. A large fraction of the world's population live in hot places, and in these hot places, agriculture uses over 90% of the water, which is a spectacular statistic. And one very important way to try to address this challenge is to use controlled environment agriculture. And I'm just going to introduce to you a spin-out company that we developed from KAUST, where we brought together plant science with engineering and material science, nanoparticles, and photovoltaics. And we've developed this company where we're trying to address this food, water, energy nexus to try to develop systems for growing plants in hot, saline environments. We've actually grown very rapidly. We're now, I think, 80 employees from multiple countries and very highly educated staff. And we have 4 main areas of focus. We've got a heat-blocking nanoparticle developed by Daria Baran, material scientist, which is turning into a bit of a blockbuster breakthrough. It's got a very sharp cut-off between absorbing in the infrared and transmitting in the visible, and has very big impacts on temperatures in greenhouses and the use of both energy and water in those greenhouses. So this is what we would call passive cooling. We have active cooling in particular, where we're using salt water to cool the greenhouse. In a normal greenhouse in these hot environments, 90% of the water used is roughly 90% of the water used is just to cool. So why use fresh water to cool when you could use salt water to cool? That's the principle there. All of this actually does require sophisticated monitoring and management of the environment, and we now have increasingly good software combined with bespoke sensors to develop a cloud-based IoT, Internet of Things style sensing and management software system, which is we're calling Cortex, and is actually becoming better by the month, I should say, and that's starting to be rolled out commercially, and it's certainly in beta testing trials in several countries, including Turkey, Spain, United States, Mexico, UAE, and Saudi Arabia. And then finally, my own genetics, where in the first target for our work is tomatoes growing in controlled environments and open fields. And we've developed a new type of rootstock. It's a series of rootstocks which are enabling the plants. It's conferring upon the plant increased salinity and heat tolerance in particular. And this is a very significant breakthrough, which we're now taking through the regulatory processes to enable commercial sales of seeds in 2024. Also developing systems in the similar type of systems. It's a new type of genetics. It's legal, it's non-GM, it's non-gene editing, but it's definitely a patentable new type of genetics, which is very exciting. We're now starting to apply that to, or develop it, I should say, don't want to get too carried away. We're starting to apply that, develop it for peppers and cucumbers and other species of plants. So the idea is for us to develop systems across the bottom here, which have much less water use. Yes.
1 minute remaining, yes.
I can wrap, yes. Thank you very much. Good, thank you. Which has got much less water use, but without any increase in energy use. In fact, if anything, we use less energy in our greenhouses. They are moderately capex intensive, but we're working on reducing the impact of our technologies on the cost of the greenhouse. But even so, the business case is still quite solid because the OPEX, the operating expenditure, is much, much lower with our systems, using less energy, using less water. So we've developed a company. We do have proof of concept commercialisation. So we have our technologies rolled out in several hectares of greenhouses that we have under our control and selling produce into the market, which is great to sort of show to customers, investors, that we actually are doing, that our technology actually works. And that's just showing that we actually do have a commercial product now, at least at the produce level. But we also have commercial products in terms of heat blocking in particular, for which sales now are really quite large and across the globe. And on that note, I'll just conclude and thank you all for listening. I do think we have lots of sustainable business models that can be developed for using saline water in agriculture. I think controlled environment systems are particularly viable and useful for local food security because of the production of high nutritional value produce in remote regions. And technologies are very important to increase the viability of saltwater-based agricultural systems. We need to bring together engineering, material science, IT, plant science, and probably a lot more. Thank you all very much.
Thank you, Dr. Mark. On behalf of استدامة Center, we thank you for this great session. And we hope to see you, inshallah, soon. Thank you, Dr. Thank you, Dr. Mark.
Okay. Thank you very much.
Thank you. ننتقل للعنوان الثالث ما قبل الاستراحة عن تحمل محاصيل الخضر للملوحة وتداعياتها العلمية. Mr. Wim Vogt from Wageningen University and Research.
This one?
No. Number 3.
Sabah al khair. Sorry, that's the only Arabic word that's popping up in my mind. I'm sorry for talking to you in English. Thank you for the opportunity to give a talk here on at the Estidama Center. My name is Wim Vogt and I'm part of the Wageningen University Research team supporting the Estidama team in their research. My talk is completely different from the previous two because it's focusing on vegetables, and vegetables are very, very sensitive for salt. So I'm talking about salt levels 10 times lower than Dr. Sayer and also Mark Tester talked about. But it's good to talk about vegetables because also at the Estidama Center we are focusing on vegetable production, and I will show you what we can achieve with, well, not saline water, but let's say less water of lesser water quality. Well, I think you all know here the relation between salt, salinity, and yield, the famous Miles-Hofmann relationship, where we have two parameters, the salinity threshold value and the salinity yield decrease, which is both parameters are very, very crop specific. And to give you just an idea, these are two agronomy crops, wheat and barley, with threshold values of 4 and 5.3 millisiemens. I'm talking about the EC in the saturation pace of soil. And the salinity yield decrease of 7.5% and 10%. But if you look to vegetables, this is, for instance, tomato, the threshold value is much lower. And also the decline is pretty high. But looking to to other crops like sweet pepper and strawberry, then you see that the threshold values are much, much lower, and also the salinity yield decrease is much, much higher. Well, I don't have to elaborate about what is salinity doing on plant growth. I think you all know, but there are two topics. One is the effect on the osmotic pressure, EC effect, and the other is the specific effect of ions like sodium and chloride, giving rise to some toxic effects and nutrient imbalances. Well, in particular, these specific effects are threatening the vegetables like tip burn, blossom end rot, and even potassium deficiency is really a threat for vegetables. However, salinity also could have a positive effect, and we even make use of that. For instance, if you grow tomatoes, then we know that higher EC levels can promote the fruit quality. And fruit quality can be in terms of the fruit shape or the shelf life, like coloring days is going down, so the fruits are more evenly ripened in a shorter period, and also the shelf life can be improved by higher EC levels. But most importantly, the parameters that affect the taste of tomatoes will be promoted by having higher EC values. So that's why we in general have this model for our EC effect of vegetable crops, particularly tomato, peppers, but also cucumbers. If you go from 0, well, that means you also do not have any nutrients, because we include, of course, in the EC also the effect of nutrients. Then you go from a low production to an optimal production. It's a plateau, and then at a certain moment, the salinity threshold level, the production goes down, according to the Maas-Hofmann model. But if you look to the effect on quality, and this is the example for shelf life, but I also could here put in the line for taste, then you can see that also here the quality improves, but the improvement goes beyond the level where the yield is already decreasing. So, well, for our, let's say, recommendation for water quality or fertilization, we have a sort of optimum EC range, and it depends on what you want to produce. If you want just quantity, well, then you go for a lower EC range. If you want to promote your product as also superior in quality, better shelf life or better taste, then of course you go to the high level. So it's more an economic evaluation what you have to do. Okay, so knowing this, then it's also important to know what is the minimum required level for your nutrient. So the contribution of your nutrients to your EC. And we did some trials in the past already, more than 30 years ago, where we found out that if you increase the EC just by nutrients, or you increase the EC partly by sodium chloride, that for the plant it doesn't make much difference. So we did these trials. You see here 6 treatments with 3 EC levels, 2.5 till 5.2. In this series here, just by nutrients. And in this series, we increased sodium chloride. And the result was clearly that we did not see any difference between the EC effect of nutrients or affected by more sodium chloride. Surely the yield was decreased, but the quality was improved, both in shelf life and in, well, we also have more blossom end rot, truly. But the main message was there is no different effect on the plant, either increasing the EC by nutrients or by increasing the sodium chloride. So from this we generally made this water quality requirement table with the 3 parameters, EC, sodium and chloride. Okay. However, you understand that the salinity effect is not an effect of let's say one moment. It's just a building up. And the building up of salinity in your system is depending highly on, of course, the type of irrigation you use, as surface irrigation or sprinkler or drip irrigation, but also the frequency of the irrigation, but most importantly the leaching rate, and also how you grow your crop, soil or soilless. So this table I just showed you does not satisfy us. Just to give you an example, what is the effect of irrigation? This is a theoretical model showing that this is the EC of the water, and this is the total quantity of water needed. And you see, this is, if your leaching rate increases, you need, of course, more water. And this is then the effect of, this is the leaching rate. But if you want 100% of yield, with, for instance, potatoes, and an EC in the saturated paste of 1.7 as the threshold value. And the higher the EC of the water, of course, you need more water to reach that level. But if you accept a little yield decrease, like say 10%, then you see that with this accepted yield reduction, you already can save a lot of water, and it can be much higher if you go, if you accept even lower yield. Then you can really save a lot of water. Just to give you some idea of what is the impact of the irrigation method. Well, that's why we need more criteria to evaluate effects of water quality, irrigation management, and also the growing medium. And that's why I want to elaborate a little bit more, to focus on a specific topic, which is soilless grown vegetables, which is now, well, in Europe, it's, one can say, the prevailing way of growing vegetables. And also in countries like in Saudi Arabia, there's a lot of interesting growing crops, soilless. Well, what's typical in soilless, it is that you have a restricted root volume. That's the most important factor. Well, of course, you don't grow in soil, clearly, but it's a restricted root volume, which means that the quantity of nutrients and water available for the plant is very, very much limited. And that means that the supply of water and nutrients should be very much adequate to your cropping system. Well, and one of the advanced systems is the so-called closed loop systems, because if you grow in soilless culture, you cannot do, let's say, deficit irrigation, or even grow exactly with irrigation exactly to the crop demand, because of the heterogeneity of your drip system, of your plants, and the very low buffering capacity. So when you want to save water and nutrients, you have to use a sort of closed loop system, or a semi-closed loop system. Oh, and then the water quality is really crucial, because you supply through your water and your fertilizers some sodium or chloride, and there's a limited uptake by the plant, so there is quite a rapidly accumulation of salts in such systems. So for such systems, it's really important to know what is the maximum acceptable sodium concentration in your inputs. If the input is higher than what you can manage by the plant, what's taken up by the plant, then you have a rapid accumulation. And just to give you an example, what can happen, this is just from a practical grower which uses rainwater in his system, but suddenly the rainwater basin was empty, so he had to apply tap water, and tap water was of a lower quality, and there was a rapid increase of the sodium and the chloride concentration, and also the EC. And later on there was sufficient rainwater again. Anyway, the sodium and chloride concentration were rapidly increased, just as an example. So let's focus a little bit on this sodium balance model, which consists of the input of water and fertilizers, where there is some water in the sodium in the water, there is some sodium in the fertilizers, and there is the uptake by the crop, which can be divided in the uptake or the translocation to leaves, stems, and fruits. In total, this is the total uptake by the plant, and there is the sodium in the cropping system. And of course, if you discharge part of the water, then there is a sodium output in the discharge, in the drainage water. What you need to realize is that if you have increased sodium or chloride levels at the root surface, then the plant linearly increases also the sodium and chloride uptake. And this graph shows you the linear effect. For instance, if you say the maximum acceptable sodium concentration around the root zone is 7.5 millimole, then the uptake for tomato is 0.5 millimole per liter. That's what we call the uptake concentration. And you can also clearly see that for cucumbers the uptake is much higher, whereas for sweet pepper the uptake is really much lower, showing clearly the crop-specific effects. So don't mix up uptake with salt tolerance. Yeah? Some plants really can take up a lot of sodium but still are not so tolerant. And we also know of plants that don't take up any sodium but are quite tolerant. So if you are able to increase the sodium maximum level to 15, then the uptake for tomato is increased to 1 millimole. And for all crops we have these equations, the linear equations, where we found that crops like roses are extremely low in uptake, and also a sweet pepper is quite low, but there is a difference in the generative and the vegetative stage of the cropping period. What is the practical consequence of this? Well, to show you this example, it's a model. It's the effect of what happens if you allow the highest sodium level in your root environment. And for instance, if you say the maximum is 5 and you have an input of 1 millimole per liter in your raw water, your source water, for a tomato 11-month cropping cycle, then this happens. You start here with low sodium, but then rapidly it increases till you reach this 5 level, and then you have to do discharge. And in the end, you need 530 cubic meters per hectare of discharge drainage water, which is also a lot of nitrogen that is lost. However, if you, with the same crop, accept 10 millimole, then the number of times you have to discharge, because you postpone the moment of discharging, but also by having this higher sodium level at the root surface, the uptake is also higher, and then the discharge needed is only 40 cubic meters, and you lose only 85 kilos of N. Okay, so the question is, what can we accept? And I again show you this model, and then we need one other parameter, and that's the minimum level we see for the nutrients. Just the sum up of, in this case, the cations, ammonium, potassium, calcium, and magnesium. What is the contribution of these 4 cations to the EC? Well, that's a minimum level to reach sufficient yield. But to reach sufficient quality, you had to increase the EC. Well, this increase in EC can be done with non-nutrient ions like sodium. So this gives you a room, a space for increase of the EC by sodium. And in figures, it's difference between the maximum acceptable EC of 4.5 of 2.8 millisiemens transferred to sodium, that's 25 millimole per liter. So that's a lot. So we did a lot of experiments in the last 5-6 years with crops to maximize the sodium uptake concentration And well, we changed the nutrient solution. You can imagine that because we want to have this minimum concentration of nutrients, and we increased from 5 till 20 or even to 25 millimole per liter of sodium. Well, I cannot elaborate too much on all the results. That's too many. It's a lot of that. This is already published. But what we found is for this range here, there were there were no significant differences in yield, and that's good news. So it means that for tomato we can even accept up to 25 millimole of sodium, and that gives that opens the possibility of saving a lot of water and nutrients, and we did that with many other crops. So finally now we have this table extended where we say, okay, you can discriminate between classes and subclasses with salt-sensitive crops, that it's not only looking at EC and sodium, but also look to in which system are you growing, in soilless, and is it hydroponics, is it with recirculation or non-recirculation, and that is the result. So come to my conclusions. We learned that crops can deal with much higher sodium concentration than commonly accepted, by, specifically by growers accepted. And sodium still can take part of your EC. It can replace part of your nutrients. So the space between the minimum required nutrient concentration and the recommended EC. The higher the EC in the root environment, also the higher the uptake is. So it opens the perspectives for lesser quality of water use in semi-closed systems. Okay, thank you for your attention.
Thank you, Mr. Wim, for this session. الآن ناخذ بريك تقريبا 10 دقائق وبعدها إن شاء الله نرجع لاستكمال باقي المحاضرات. نبدأ الآن المحاضرة التالية بعنوان تطوير حزمة عروض القيمة للمحاصيل الملحية الموجهة للاستخدام الغذائي خارج الطريق لنهج المستدام من المزرعة إلى الشوكة. والدكتور دونيسيا from إكبا. دكتور دونيسيا. Good morning. Yeah, it's mute.
Hello.
Yeah, hello, hi. Hi,
Hello.
hi, good morning. Welcome to our session. So please, can you start now?
Yeah, sure.
Yeah, you have 20.
Share.
Yeah, share it, and you have 20 minutes for the session. Please.
Okay, thank you so much.
Thank you. Please. Go ahead.
And the team for inviting me to attend this workshop, to participate as speaker, and also to listen to these very insightful presentations from the previous speakers on halophytic and salt-tolerant varieties of conventional crops. So, can you see my screen now?
Yes.
Okay. So, is it okay? Hello?
Yes. Yes, it's clear.
Oh, okay. So, my name is Cici Lira, and I'm working as a halophyte agronomist at the International Center for Biosaline Agriculture. For those of you who are not familiar with our center, or else ICBA, the acronym of our center is ICBA, we are focusing on how we can develop climate resilient and sustainable farming systems in hot and dry areas and overall salt-affected areas and marginal lands. So my presentation is about the development of the value proposition package of halophytic crops that we are working on, directed for food use. And I will show you this package consists of different stages to have in place a sustainable farm to fork approach. So as the previous speakers have highlighted, we have in a salt-affected environment, in a salinized area, we have two options to move forward with the farming systems. Either to use salt-tolerant varieties of the crops that we know, of vegetables, or in higher salinities to focus on farming halophytic species. So some of the food and feed crops that we've been, conventional crops that we've been focusing at ICBA, you see the list: date palm, pearl millet, finger millet, sorghum, potato, rice, and from as feed crops also buffalo grass, blue panicum. And from food perspective, the halophytic crops that we've been working on is Salicornia, ice plant, civet, New Zealand spinach, and from feed, for feed use, we have Sporobulus virginicus, Arabicus, Paspalum, Naipa grass, Disticlis spicata. So just for you to have an idea, what is actually a halophyte, right? So halophytes, and this beautiful graph developed by Mans and Mark Tester, really explains that halophytes, in comparison with the conventional crops, really grow in very high salinities. As Dr. Afif mentioned at the beginning, the halophytes grow beyond 200 millimolar of sodium chloride, and we see that tall wheatgrass and saltbush, they really produce high biomass under these high salinities. So the market proposition, the value proposition for the salt-tolerant varieties of the conventional crops is already known. The consumers, the public, are already familiar with the use of carrot, the use of salt-tolerant potato, cabbages, lettuce, kale, whatever. However, when we move to the halophytes, most of them, the majority, I should say, of the consumers, they are not familiar how they could utilize these crops. And of course, apart from the cultivation practices, what actually the uses that they could be in place. For the last 10 years, ICBA, we've been focusing on Salicornia bigelovi research. Salicornia is a succulent halophytic plant. It's an includer plant that really absorbs the salts from the medium where it grows, and it has three main benefits. First of all, it is a multi-purpose halophyte, so it can be used for food, as feed, for industrial, pharmaceutical purpose. It can be irrigated with high saline water, even with seawater. Actually, its natural ecosystem where it naturally grows, it's coastal areas, tidal areas, marshlands. We can even utilize brackish groundwater. At ICBA, we've been focusing on how we can utilize the byproduct from desalination, the reject brine, aquaculture effluents. And of course, the specific species, as Professor Mark Tester highlighted, it is well adapted in hot and dry areas since its nativity was introduced in UAE, of course through a collaboration with Masdar Institute in UAE in Abu Dhabi and with CAUS. So they brought some germplasm collected from the western, southwestern Mexico, where we evaluated different kind of ecotypes that were collected. So this species, this Salicornia species, is very well adapted in hot and dry areas. So initially, Salicornia bigelovi and the whole research initiated because Salicornia bigelovi, exactly the seeds of Salicornia bigelovi, they have really high oil content. It ranges between 26 and 33%. So it shows some good potential to produce biofuel. However, when we are actually going deeper in making the calculations, and based on an anonymous reference, 1 hectare of Salicornia bigelovi can produce approximately 675 to 750 liters of biofuel. So consider a 1 hour flight of this Airbus plane, let's say from Delhi to Mumbai, running in a speed of 600 kilometers per hour and the capacity of 192 people. It consumes for this 1 hour flight 5,000 liters. Of course, we have to consider that the biofuel and whatever attempts have been done so far, they have used Salicornia biofuel as a ratio, not a flight full dependent on the biofuel originated from Salicornia. But considering that we have these 5,000 liters covered by Salicornia, it means that we need 7 hectares of Salicornia bigelovi's biomass and of course seeds to have in place. Which of course this is translated to huge production of Salicornia bigelovi seeds and massive land that has to be cultivated. Of course, when we're talking about land, like desert areas, coastal desert areas in Middle East, of course, the number is not that big issue. However, there are some other challenges that we have to consider, especially for biofuel production. First of all, Salicornia bigelovi is an annual plant, as it was already highlighted, and for having a feedstock to be directed for biofuel, we need to have year-round production. So for the current being, this cannot be achieved, which means that we have to have additional feedstock for biofuel apart from Salicornia bigelovi. The other hindrance, the other obstacle is the small size of the seed. As you see in the upper photo, the Salicornia, the size of the Salicornia bigelovi seeds is just, it ranges between 1 to 2 millimeter, and it's really hard to have sophisticated machines in place to harvest and collect the seed as clean as possible. Of course, the other, let's say, challenge is, especially when we are growing Salicornia in a coastal area, and of course there is this high risk of salinizing further the soil, is how we can actually manage and to alleviate the soil from the salinity buildup. Of course, there are practices in place like proper drainage, probably proper leaching fractions that we can actually have in place in order to alleviate and control the salinity buildup. So apart from the biofuel and of course the oil aspects, definitely Salicornia bigelovi, as it has been stated in references and literature, can be also used as vegetable oil. However, from other industrial uses point of view, there has been a lot of focus now on Salicornia bigelovi for biogas production, for bioethanol. Also, how we can utilize pyrolysis as a kind of method that would improve the biofuel production, biorefining, and other kind of processes. The second use that Salicornia bigelovi is also a useful plant to consider is that it can be used as feed, can be incorporated in the meal of animals' diets. And when I'm talking about animals, it's mainly small ruminants like sheep, goats, and camels. So these are some of the data that we have come up with from different kind of analysis here. So you see the comparison between fresh Salicornia, dry Salicornia, and mixed Salicornia. Of course, and I have also included the range for the crude fiber, the acid detergent fiber, and for the neutral detergent fiber. So you see, when actually we are mixing Salicornia with 25% of Salicornia with 75% of forages, definitely overall the parameters, the quality, the nutritional profile of this feedstock definitely is improving, and of course the salt content is decreasing, which is a high risk factor to consider, especially you see for dry siliconia it's really high. So when we're actually using it in a mixture with other forages, it can significantly drop. So this type of mixtures can be usefully utilized for animal as animal feedstock and as animal meal. So the Salicornia bigelovi can be mixed up to 30%. This is the recommendation also from the literature and from the studies and from the research conducted so far. The third use of Salicornia, and you will see different versions, and of course I'm going to focus more in the next slides, and especially at the end of my presentation on the food side. So just to show to you what kind of interventions we have done in terms of directing our focus for the food use for Salicornia, and the different versions and the versatility of Salicornia being used in different food products. So the second step of the second stage of our work is to evaluate the Salicornia bigelovi germplasm. So we have done different field trials, and definitely we've been focusing on utilizing the fresh tips, which are directed for food use, then on the green biomass yield directed for forage use, on the seed yield, definitely using different salinity treatments and different water treatments, starting from brackish groundwater, the reject brine from desalination, and the aqua brine, where we actually, because we ran a think about the integrated agri-aquaculture system, where we direct the reject brine from desalination to grow tilapia. So here you can see some yield parameters, some yield data actually that we have achieved after using these saline water treatments. The same applies for the green biomass and the seed yield, and data have shown so far that aqua brine, exactly because the fish add this fertilizer component through their waste, it's what we call aquaponics. Actually, it's saline aquaponics, favors more biomass production compared to seed yield. We have also run a coastal farm in Quain, in an emirate here in the United Arab Emirates, where actually we have tested different Salicornia genotypes being tested with the aquaculture drainage from a marine environment research center, and also partially water coming from different fish species like sherry and tilapia. So you will see here in the photo below that here the Salicornia is much more grown compared to the Salicornia in the upper photo. The difference is that here we've been irrigating Salicornia with the drainage coming from the whole marine environment research station, which was flowing into a mangrove area, compared to separately sherry and tilapia, compared to aquaculture effluents coming from sherry and tilapia tanks. Definitely throughout all these years experimentation, we've been focusing on how we can improve and how the different genotypes they perform in terms of green biomass and seed yield under different salinities, and also under different environments. For example, we worked on a project where we implemented and introduced this integrated model of Salicornia plus the fish and Salicornia alone in 8 farms in Abu Dhabi Emirate. You see here the Salicornia growth in different farms. Of course, there were challenges in place that all these have been, most of these actually have been included in a chapter that has already been published. You can find it here. And you see the really high saline, the high saline content of the water that was used, and of the groundwater and the reject brine that was actually utilized for the fish and Salicornia farming inland. The second part of the germplasm evaluation is the germplasm improvement. So for the last 4 years at ICBA, we have been targeting to inbreed the current germplasm in order to create homozygous kind of lines where we can move forward with genome sequencing. And the second approach is crossbreeding for population development.
Dr. Denisia?
The breeding of them.
Doctor,
Yes.
remaining 2 minutes remaining.
Yeah, okay.
Please.
So the breeding objectives, of course, is is to improve the biomass and the seed yield, and definitely throughout all this breeding work, we have been focusing and we have come up with some bunchy types, early maturing tender plants, which were actually utilized for this fresh tips harvest, right? The third package that we are focusing on is the management practices improvement. This is a package that we've been focusing on fertilization, different irrigation systems like using drippers, bubblers, subsurface irrigation for Salicornia, cultivation, planting density, and mechanization. Because as I mentioned, there are no customized machines for Salicornia, for fresh tips, or for even the seed collection. So this is something that we have to tune in and see how existing machines can be customized for Salicornia farming. The other question that we've been very frequently addressed is salinity building up in the soil? As I mentioned before, there have been these farms where actually we observed that there was a kind of buildup, salinity buildup in the soil even after the first season. The graphs that I am showing are actually from the salt leachate experiment that we've been and project that we've been working together with different eminent partners from UAE, like the Environmental Agency in Abu Dhabi, and from New Zealand. So actually what the results show is that when we are using a bubbler system, drippers and subsurface irrigation system under sandy soil, so the sandy soil here it's around more than 95%, and of course using proper leaching fractions, we observe that there is no actually salinity built up in the soil profile below 40 centimeters. Of course, this is something encouraging. However, we have to be cautious because we are really irrigating with very high saline water that definitely adds more salt in the groundwater. The fourth package is the nutritional value proposition. So we've been working on intensively the last 2 years on how we can, at different growth stages, how we can focus and utilize the fresh tips. So this is why you see early, mid, late vegetative stage, and also the comparison with spinach and asparagus. We have done extensive nutrient profile for micro, macronutrients, for amino acids, fatty acids for both fresh tips and seeds. So these are some of the results. So the early vegetative stage of Salicornia miguelovi is rich mainly in vitamins and amino acids content, whereas with the progress of the plant growth, the plant increased its nutritional quality in terms of fiber, carbohydrates, minerals, vitamin C, and vitamin B3. The early vegetative stage is mainly directed to be consumed as fresh salad green. However, the older vegetative stage can be explored in processed food products due to the increased fiber content. And also, we are currently exploring the Salicornia seeds for food for edible uses apart from the oil use. Regarding the economic viability of Salicornia, return of investment, so these 2 slides, they are actually based on these 8 farms. We've been actually deep diving now into more, to tune in all these different economic scenarios. Salicornia has a program in place. However, we have to consider that we have the proper management practices and proper package in place. The market proposition. So what consumers are expecting from us, even if we have the perfect package in terms of management practices, the varieties, whatever. Yes?
It's time.
I will finish.
Yeah, please.
Yes, okay.
1 minute, please.
Yes.
Please.
So the question is what actually we can use with these crops, with the Salicornia, how it can be used. So we've been working closely with chefs from fine dining cuisines, or even for food security, because you see from food security perspectives, because Salicornia can be used as ingredient in different kind of forms, in solid, liquid, also in sweet and savory dishes, which really has expanded these Salicornia uses for food. We've been working with a food company, Al Arish, in UAE, it's based in Sharjah, where actually they sold a Salicornia-based burger patty, where Salicornia was used as ingredient. Also, Salicornia has great potential as ingredient in extruded cooking products. You know very well that on local level, on country level, there is high interest, especially in Middle East region, to develop the cuisine portfolio, what can be locally produced and farmed, so that it can be inserted in the local dishes. We have worked closely with partners in Egypt and Morocco, where we have introduced Salicornia. Last year, we received an award for this project proposition. And the conclusion is that the halophytes can definitely play a role in salt-affected areas. We have to focus in order to have the proper ROI, return on investment, and multi-purpose use. Definitely, a multidisciplinary approach is needed in order to have the value proposition in package in place, which takes more than 10 years in order to work on that. The food industry can play, food and feed, I should say, can play a huge role in halophyte and leveraging halophytic innovative uses. And of course, for such kind of projects, we need efficient leadership with really strategic mindset in place. I would like to thank my team, and of course, again, Estidama team for having me on board. Thank you so much. Thank you.
Thank you, Dr. Tunisia. On behalf of Istidama Center, we thank you for this great session, and we hope to see you, inshallah, soon. Thank you. Thank you for joining us.
Thank you.
لننتقل للعنوان التالي بعنوان التخفيف من ضرر إجهاد الملوحة في الطماطم عن طريق إضافة البوتاسيوم مع اثنين من المتحدثين من منظمة الفاو، دكتور إحسان الحق and الدكتور عبد الله الهندي.
Bismillah ar-Rahman ar-Rahim. My name is Dr. Ihsan al-Haq, and I'm working with the United Nations Food and Agriculture Organization in Saudi Arabia as an international value chain expert. First of all, I'm very thankful to Istidama for arranging such a powerful discussion here. Our discussion today is divided into two parts. First, we'll have a look at how this salinity is affecting tomato productivity. And then in the later part, my colleague, Dr. Abdullah Al-Hindi, he will discuss the salinity profile in Kingdom of Saudi Arabia and how it can be alleviated. Our discussion will go like, first of all, we'll discuss some salinity stress, how salinity damages the plants, its physiological impacts, sodium-potassium balance, then what mitigation measures can be taken through potassium supplementation. Then salinity in KSA will be discussed by our colleagues, salinity standards and international and local both standards. So having a look at some of the basics, we know that salinity can be caused by the presence of many salts, but sodium being the biggest culprit in that. And with this salinity comes salinity stress, which actually translates into reduction of production plus quality, and that is reducing the potential of our crops to feed the increasing population. This is one thing. And it is adversely affecting UN target of reaching the zero hunger. Besides that, it is also translating into loss of millions and millions of dollars throughout the world. So if we look at different crops, vegetables, fruit crops, field crop, and forage crops, these can be divided into different groups like tolerant, moderately sensitive, and sensitive crops. So those which are tolerant, there is less issue dealing with the salinity. But unfortunately, those are not much in abundance. So we need to find ways how to deal with the salinity, how we can mitigate their bad effects. So we need to first have a look at how this salinity actually affects the plants. Basically, whenever we say that there is high soil salinity, it affects the plants in two different ways. One is it creates some metabolic issues. The other is it induces cytoplasmic toxicity, all relating to ill effects. So dividing these into different categories, we know due to high soil salinity, both chlorine, sodium, and boron, these gets into the system and excluding some of the very much required nutrients like potassium and calcium. These are thrown away from the plant system, resulting in induction of nutritional deficiency. And this has been alluded to some of my previous presenters. So, and this also induces some water deficiency symptoms because of osmotic imbalances caused by high salinity, mainly again sodium. The soil water issues relates to hypoxia and ion leakage from the cells. Hypoxia again leads to low respiration by the roots, and ion leakage actually is a compromise on the basic structure of cell. So what it does is total quality loss and reduction in the production. So what mitigation measures can be taken to alleviate those ill effects? Our colleagues, they have alluded to some of those mitigation measures like genetic improvements, like agronomic practices. But we think that there is no such thing as a silver bullet. There is not single solution to it. A combination of many activities that will lead to effective alleviation of ill effects of salinity. So like if we talk about agronomic solutions, fertilizers, nutritional, soil reclamation, bioremediation, these are some of the actions that can be grouped under agronomic facilities. Some of the researchers, they have used efficient phytoprotectants like melatonin, amino acids, plant growth regulators to mitigate the ill effects of salinity. We can also utilize biofertilizers like mycorrhizal interactions with the soil. Those can also alleviate the ill effects of soil salinity. And of course, breeding through conventional and non-conventional method for varieties, for crops which are tolerant and resistant to increased levels of soil salinity. In short, a combination of all these three, depending upon the situation, will result in reducing the ill effects of soil salinity. So if we have a look at the cascade of salt stress effects, we know that when high chlorine or sodium is taken up by the cells, few things happen. When it enters into the cell, it really, results in physiological drought, which leads to decreased osmotic potential and making the cells flaccid. It actually results in compromised shelf life. The other is when calcium, chlorine, and sodium is high in cells, it disrupts the metabolism of that system and disruption of organelles, which again results in decreased yield and growth. Nutritional imbalances also occur at the same time because when sodium and chloride ions are taken up by the plant, it replaces some of the nutrients in the soil. So what the plant does, plant has two different strategies to combat this. The strategy one is that it tries to go to ionic homeostatic thing and tries to maintain the balance of it. So we know that within the cell, if the concentration of sodium gets higher, when it enters into the plant system, into the vacuole actually extrudes some of the hydrogen ion or the protons into the cytoplasm. When the proton or hydrogen ion concentration in the cytoplasm increases, it leads to cytoplasmic acidosis. When the acidity of cytoplasm increases beyond certain level, the enzymes present in the cytoplasm, they started dysfunctioning, damage to different organelles occur, and resulting into cell membrane damage. So, second strategy by the plant cell is extrusion of these sodium ions from the cell. But it also has a cost, both energy cost and it requires transformation. When the sodium ions are extruded, it takes in 2 potassium ions for 3 sodium, repelling 3 sodium ions, which requires energy. Another ill effect is that this sodium goes into extracellular spaces, which leads to physiological water deficit and disrupting the cell wall structure. The energy utilized by the plant in doing so again leads to an imbalance between source and sink, which leads to lower yields. This experiment we conducted a few years ago in Pakistan in the controlled environment. We applied different potassium fertilizers, mainly muriate of potash and sulfate of potash at different salinity levels. So we found that with the increasing soil salinity levels, the yield decreased significantly, reaching up to 1.15 tons per hectare. This is the minimum yield. And even the highest yield at control, it might be lower because it was not a hybrid variety. So, okay. So, and again, at different salinity levels with different potassium applications, muriate of potash and sulfate of potash, we found that the leaf sodium content, it consistently increased. The potassium content, it was decreasing with the increasing salinity levels. And most importantly, the proline content in the leaf samples, it was also increasing with the increasing salinity levels. When the proline accumulation is higher in the cell, it can lead to asphyxia and ultimately death of the cells when it goes beyond certain limit. And another important aspect was that with the increasing salinity levels, the membrane leakage, which was measured after 30 minutes, was also increasing, leading to the conclusion that increasing salinity levels actually leads to a compromise on cell wall structure. And in both the cases, you see here that the sodium content in the leaf tissue, potassium content, it was being reduced. And the leaf proline content, it was going up. Cell membrane leakage in the leaf discs, it was also going up. But if you notice that with the sulfate of potash, it was a little bit lower than with the muriate of potash, probably due to muriate of potash having chlorine, containing chlorine ion. So the conclusion of our experiment was that salinity basically decreased both the fresh and dry weight of roots and shoot, by increasing the sodium concentration within the cells with a concomitant decrease in potassium ions. It also altered the balance between sodium and potassium ions, while it induced decline in growth and yield, whose ill effects could be reduced by the application of supplemental potassium to tomato plants. And among the potash sources, as I mentioned earlier, sulfate of potash was more effective in increasing root and shoot fresh weight and dry weight, as well as yield, besides decreasing the membrane leakage. However, sulfate of potash was less effective in increasing proline content of the shoot. This low and slow increase in proline synthesis with sulfate of potash treatment, this may indicate that SOP was effective in decreasing the adverse effect of salinity. Lastly, the yield data revealed that sulfate of potash is partially effective in decreasing the salinity-induced damage in tomato crop. So with this, first part of this discussion concludes, and now I'll request my colleague, Dr. Abdullah Al-Hindi, to come to the roster.
This way, Hassan, this way.
Yes, yes, this is next, this is previous, this is next.
This is next. السلام عليكم ورحمة الله. أسعد الله صباحكم بكل خير. I was requested by the FAO Saudi Arabia management to deliver a presentation after the presentation of my colleague Dr. Hassan, and frankly I was told just I have only 4 slides. So before I go to my slides, I have just to say that my experience about irrigated agriculture in this country span over 35 years. I have dealt with all irrigated agricultural regions, starting from the southern part of Saudi Arabia, Wadi al-Dawasir, then going to Al-Kharj and Harad, moving to Al-Washim and Sajir, and then to Al-Qasim, Hail, Al-Jawf, and Tabuk. Before I retired from the ministry, I was the team leader for a project related to land degradation. And our focus was on 2 major things, and that's salinity and fertility depletion. So it is not a scientific paper, what I'm going just to show, and it is only based on a very practical way of a big survey. It is a huge project. We planned at that time to see what's the situation of salinity and fertility in all regions. Unfortunately, we were able just to deal with Wadi al-Dawasir and Hail. So just to show you, okay, these are the 2 documentations of our work, and we had to analyze hundreds of soil samples in each region. And for soil samples, we specified 16 parameters. And then we analyzed soil water, hundreds also of soil water, and we dealt with about 12 parameters. What strikes me when we were dealing with these truffle missions in all regions, that the farm managers and most, if not all, farmers, they know nothing whatsoever about the classification of salinity, water salinity and soil salinity as well. So in that, I thought we will have some people here who are dealing with the farms. I never ever thought that we are going just to deal with the scientific community. But in my judgment, I will just refer to the International Center of Irrigation Water Classification, and just to specify a personal experience, if you see the last one, not suitable for cultivation, and that was stated long time ago in handbook number 60, that if your water is more than 3,200 parts per million, this is the units which are known and dealt with in this region at the moment, you cannot cultivate. Last year, I was in Qasim, to be very specific, in Al-Fawara, and this is a small area in the northern part of Qasim, and they are cultivating wheat, local variety, named Muayya, and this Muayya has two different, if you like, land rays, Hilba and Samma. And I was surprised to see the irrigation water was more than 3,200 parts per million. And that farmer was able, it is a very well calculated statistical figures, to get about 5 tons per hectare of that very specific local varieties. So that is, if someone is in the future thinking about official breeding program, I think really we should look to what we have, rather than jumping to halophytes or something else. This is not just to criticize what we have seen, but this will simplify the system that we can deal with. This is, and I am very much willing even to have any kind of interaction with anybody if someone is willing for that. So now I intentionally put the local standards of soil salinity classification. Throughout this soil survey, huge soil survey, we have come, you see moderately saline soils. Once again, if you go to the literature, it is very hard really to cultivate. That is what the literature is telling us. If your soil solution, the electrical conductivity of your solution is more than 4 decim per meter. The reality, if you just go to this section, we have seen in Hayy, I have seen myself, the salinity of that field is about, yes, it is a soil compost. And maybe it is not very representative of the whole field, because the field is about 50 hectares. But that farmer was able to get 7 tons per hectare. Although we analyze all the samples, including the compost samples, and the salinity was almost 7 decim per meter. So if we look to those very critical issue of salinity survey, we may come up with a certain conclusion that we have really to deal in establishing a breeding program. By the way, this country does not have an official breeding program up to now, either for vegetables. Most, if not all, seeds are imported. And also we are here, if we come to the field crops, and to be very specific about the most important field crops, and that is wheat, we still cultivating Kororogjo, which is breeded 40 years ago by Semet, and that is not really a modern agriculture if it comes to the field crops. Okay. Just dealing with these things, I felt really it is very important for farm managers, and also for educated, if we can, educated farmers, to deal with such things in order to understand the severity of salinity. This one could not be fixed. It might be changed in the future. You may either expand every single part of it or shrink it. So the ultimate conclusion, we need farm managers and farmers who really understand the issue of such very critical figures of salinity and its effect on field crop. Now, this is very recent. I hope this is can be read. I'm not sure whether it is clear or not. This is in Jazan. If we just look to the, we have only. I'm not sure. Do you hear me? My voice is quite clear. Yeah, it's okay. It's okay. So in Jazan, the source of irrigation, well, let me say this. In the southern part of Saudi Arabia, we could have a very flourishing, sustainable agriculture there. I am, my job with the FAO, I am responsible, I am the MPO, National Professional Officer. I am responsible for rain-fed cereals. And what I have seen there, that the sustainability of cultivating cereals in that region, the southern part of Mecca, Al-Baha, Asir, and Jazan region is a reality. It is very visible, and we can really produce some good cereals there, even if it is not a huge amount, but it can be good for the local people and good enough. So I knew about the long time ago about this, in what way those people are irrigating and from what source. Okay, just one minute. They have surface aquifer, and it is adjacent to the sea, and the seawater intrusion is doing its part now, and it may really pollute all of the aquifer from the Yemeni border up to Al-Gunfudah. And I did this survey last year. It's not really covering all the region, but I dealt with the major governorates of productivity of producing agriculture there. And if you see here in Jazan governorate, where is the capital located, is almost 3,000. And if you go and talk to all MIWA, the people of the ministry, about the seriousness of the salinity buildup in the aquifer, they don't really believe it. And I'm afraid the scale of building up salinity, and that is because we pump more water than what can be replenished. And the issue is my belief is to deal with survey and see what's the situation on the ground, and then take your action really to improve the whole situation. Thank you very much.
شكرا دكتور حسان. دكتور عبد الله يعطيكم العافية على السيشن المميز. ننتقل لآخر متحدث معنا وسينضم الآن معنا بعنوان ابتكارات الزراعة الملحية تعزيز الأمن الغذائي والاستدامة. دكتور محي الدين عمر أهلا دكتور.
أهلا بحضرتك. Can you see my screen now?
Yeah, please. So welcome Dr. محي الدين. You can start now. You have 20 minutes for the presentation.
Okay. Now today I have been in a field trip, a very urgent field trip, and I'm presenting now from Akar in a very remote area. And I hope that the internet connection is very good. But I was supposed to be in this field trip yesterday, and it changed today. And it's my pleasure to be here today. Never slept. And today I'm going to present our efforts with regard to saline agriculture environment. So at ICARDA, firstly, I'm Muhi Omar as a research associate at the Soil, Water and Agronomy team at ICARDA. So we have several contributions to saline agriculture. So let me very quickly, I know that you have already so many informations about the saline areas worldwide. As you know that currently is about 833 million hectare worldwide. This in addition to many areas in the world that still have missing information about soil salinity. This according to FAO. And for sure, as you all know that in the future, the soil salinity and the areas of soil salinity worldwide will increase due to the climate change, saltwater intrusion, and unsustainable agronomic practices. Usually, okay. So usually there are, sorry? Okay, so usually there are two main reasons for soil salinity: natural and human-induced. Natural, as it's called, primary salinizations, usually in arid and semi-arid areas where evaporation rate is more than precipitation. So water usually evaporates and leaves salts accumulating in the soil. And also there are some other natural reasons such as lakes and areas near and close to sea. On the other side, there are some human-induced reasons. First reason for sure, when we are using irrigation water of high salinity. Also another reason is removal of deep-rooted vegetation, which plays a very significant role as drainage. Also over-irrigation, because over-irrigation usually is some kind of salt loads, increasing salt loads in the soil, and also over-fertilizations. Lack of drainage also is one of the reasons for soil salinity, and intensive drainage used in some areas. For example, they are depending on drainage water used as to compensate the lack of fresh water resources. Also, for sure, water pumping and the coastal plains and the climate change, mainly due to sea level rise and saltwater intrusion. So we have several contributions to control and adapt with soil salinity and saline agriculture. So first one is microtopography-induced soil salinity control during leaching. Actually, this intervention or this innovation is where the soil is saline, but the water is fresh, or the water is of low salinity. Actually, this can be found in many areas. Here is an example of this area in Sahl al-Tina area in North Sinai in Egypt, where a depression is surrounded by both Mediterranean Sea from one side and Suez Canal from the other side. So the soil in this area is saline, but the source of water is fresh and is of good quality. The source of water is Salam Canal as a mixture between Nile River and drainage water from Bahr Hadouz drain. So flood irrigation water is predominant everywhere in this area, where the water, as you all know, is pumped from one side and then covers the entire field and then is being drained from the other side. In this case, and due to the irregularity of flood irrigation, so the soil is being salinized in some areas and is being leached in some areas. So there is not a good or well leaching procedures that have been taken in this area, and that's why the microtopography induced soil salinity control during leaching is to install internal ditches and drains, as you can see in the figures. So between each two ditches, you have to dig a deep drain. So by this way, you improve the leaching efficiency. So the water is moving from ditches to drains by two ways: one laterally or horizontally through the banks of ditches, and the second way is vertically, because the drain is very deep, so it can absorb any water that coming vertically from the ditches, and in the same time can absorb the water table that is rising with tide. So this way, or this topography, this microtopography method has been applied, and as a result, the drainage salinity reduced from 37 3 times to 10 times. Usually, the range of the drainage water salinity or the range of the ratio between drainage water salinity to irrigation water should be between 3 to 10, according to the salinity of the irrigation water. So, and the drainage water salinity also is an indicator of soil salinity, because usually it collects and absorbs soil salts. So in this case, and after 76 days, the drainage salinity decreased to only 10 times of irrigation water salinity. So, and soil salinity also decreased from about 12 to 2.5 decimens per meter. Using accumulated water height 104 centimeter, which falls within the range of acceptable leaching fraction. Also, we have been contributing to reducing irrigation water, because as you all know that irrigation water is a source of salts by the end. So if we are over irrigating or not irrigating precisely, so there might have been some salts accumulating in the soil. So we are reducing irrigation water by precision irrigation scheduling and improved field application efficiency. So by precision irrigation scheduling, we have been introducing and offering new innovations such as thermal imaging based irrigation, soil moisture based irrigation, and accurate evapotranspiration. And on the other side, we improve the field application efficiency by introducing improved gated pipes and lightweight raised bed machines. So for the precision irrigation scheduling and the approach of thermal imaging based irrigation, based on approach that when the soil moisture below the threshold of irrigation or when there is water stress, the plants start to close its stomata in order to reduce evapotranspiration. So in this case, as evapotranspiration is the main reason for cooling, so in this case the temperature of leaves increase. So we have been detecting the difference between leaf and air temperature in order to get the threshold to start irrigation. So we have been proving this concept in different locations in Egypt with different crops under different agroecological conditions and with different irrigation systems. And here you can see just an example of some results when the irrigation water amount declines, the temperature of the leaves increase. Here also you can see the graph which introduced a significant relationship between the difference between temperature of leaves and air on the horizontal axis, and on the other side between the relative water content of the leaves and soil water content. And here by these graphs also you can see the threshold to start irrigation in case of wheat in new reclaimed areas in Egypt, as just an example. Also, we introduced soil moisture based irrigation based on soil moisture readings that has been transferred to our control panel. This technique should consider three main factors. The first one is the soil texture, because soil texture playing an important role in determining the soil moisture levels, mainly field capacity and permanent wilting point. The second factor is crop type, because based on the crop type, the readily available water will be estimated. So, but usually from our experience, field crops such as faba bean, wheat have the same readily available water. Also, as the third factor and the last is the growth stage, because at each growth stage we have to set the soil moisture at a certain depth. This also just a scheme of how each factor affects the selection of the threshold to start irrigation and the depth where we have to set the soil moisture sensor. From our experiment in Uzbekistan, we have found that based on this system, we saved from 30 to 35 of irrigation water compared to traditional irrigation scheduling schemes in Uzbekistan. Also, we have been establishing and operating an evapotranspiration network in different areas in the MENA regions, in different countries in the MENA regions, in order to test the most suitable method for estimation of crops water requirement. We have been comparing different methods, and at each country, at each area specifically, there are some methods that can be recommended for precision irrigation scheduling. On the other side, as a way to reduce the irrigation water to the field, we have also improved field application efficiency. We have already developed a raised bed machine that can organize and can dig ditches as furrows that can be used as furrow irrigation, and also this machine can disseminate the seeds regularly on the raised beds. And also this system, for sure, compared to flood irrigation, saved about 25% of irrigation water, and also increased grain yields, and increased the overall water productivity, and saved about 50% of seeds used for planting. From our experience in many countries, specifically in Syria and in Egypt, we found that introducing lightweight raised bed machinery is more scalable in terms of most of farmers in these areas are smallholder farmers, and such big machines needs tractors that higher than 35 horsepower, which are not common for smallholder farmers. So now we are introducing new farm machinery for raised bed machinery. Also, with regard to furrow irrigation, as we have found that is more productive for smallholder farmers, we also improve the gated pipes for better furrow irrigation. So we have been introducing different types of gated pipes that has absolutely improved the flow of water via furrows to reach the field's ends in much faster time. It reached sometimes quarter of the time compared to the traditional furrow irrigation. Also, it reduced the seepage and evaporative losses from the earthen irrigation ditches. Also, we have some interventions with regard to reduction of evaporation, because as you know that once the water evaporates, it leaves salts accumulating in the soil. So we want to reduce evaporation properly. So we have been introducing a new climate smart agrivoltaic systems, which based on solar panels that can shade the field crops partially in order to protect the crops from heat stress and also reduce evaporation. And in the same time, the solar panels can collect the evapotranspirated water from crops, and this makes some kind of cooling for panels, which increase and improve the efficiency of solar generation. We have been introducing that currently in 5 governorates in the Nile Delta in Egypt. Also, as you all know, mulch is very important, specifically with drip irrigation to reduce evaporation. And also from our thermal imaging detection of water stress, we have found that using mulch also increase the potentiality of plants to grow under drought and saline conditions. Also, we have been working on addition of soil amendments, not to reduce salinity, because the reduction of soil salinity is a very complex issue, but to improve the performance and the water productivity under saline conditions. So biochar has been tested and investigated. Biochar by composition of plant waste. So we have been producing biochar from maize residues in a desert area in Shandawil in South Egypt. And also we have tested both fresh water with salinity of about 300 ppm and saline water with salinity of 3000 ppm. And we have found that biochar has increased the yield with both with fresh water and with saline water. For example, biochar has increased the yield in case of fresh water from 3.7 to 4. And in case of saline water, increase the yield from 3 to about 3.5 ton. Also, we have found that basalt is a natural material that can be used as soil amendments and improving the yield, increasing the yield under saline condition. Actually, basalt has been extracted from a mountainous area in the southwest of Egypt, in a desert oasis called الواحات البحرية. And this basalt, as you can see in the figure on the left side, this basalt that has been grinded from the mountain. And in the middle picture, this is a grinder that can make these small particles into powder that can be disseminated into the field. And by mixing basalt with different ratios of compost, we have found different water productivities and different yields for different crops. Also, crop diversification with conservation agriculture is one of the interventions that we are focusing on to improve the yield under saline conditions. And as you all know, that package of diversification and conservation agriculture consists of minimizing soil disturbance as possible and keeping residues, and also keeping the crop rotation from one season to another, and also keeping intercropping. So we have found that with some experiments in Uzbekistan, that such package of interventions of crop diversifications and conservation agriculture has increased the yield, the net profit, has increased so many parameters and indicators. But the most important for our presentation today is that it decreased the soil salinity by 7%. Lastly, as I have seen in one of the previous presentations, salt-tolerant crops are one of the alternatives that could be introduced in case of saline conditions. So ICARD has a gene bank, was previously present in Syria and then has been moved to Morocco. Actually, this gene bank can introduce different varieties and different genotypes that resilience to salinity stress. Our breeders and our experts are working with different crops, faba bean, wheat, barley, and different other crops. So, and from time to time are introducing new varieties that are being tested under different circumstances of different countries and different agroecological conditions, and then certified and then can be introduced to farmers. So in conclusion, as you all know, that there are two main reasons of soil salinizations, man-made and natural-made. Also, we have been working in microtopography induced that has proven its for soil salinization control and mitigation. Thermal imaging and soil moisture based and evapotranspiration approaches all are methods that can help in and can contribute to precision agriculture and precision irrigation scheduling, and that's why all these methods should be considered to reduce irrigation water and subsequently salt load. Also, some farm machinery like raised bed machinery combined with improved gated pipes are also some agronomic practices that can be taken into account in order to reduce the irrigation water. Also, agrivoltaic system and mulching from our investigations, effective interventions to reduce evaporation on the farm level, conservation agriculture and diversification of crops, soil amendments are all interventions that can be used for salinity control and salinity management. And finally, salt-tolerant varieties can be developed, and these all my interventions, and thank you for your listening. Thank you for your attention.
Thank you again. أشوفكم على خير بإذن الله.